Okay, so I work on, I'm a microbial ecologist, and so that means I study microbes and how naturally occurring microbes in the environment function and also interact with abiotic and biotic components of the environment. And so the title of my talk is Trait-Based Aerial Dispersal of Arbuscular Mycorrhizal Fungi. Many of those things probably don't have a meaning to you, but I think I'll define them as we go. Okay, so I study microbes that live within what is called the plant microbiome. So you might have heard of the human microbiome, right? So microbial organisms that live in our bodies and on our bodies, and they affect our health. Well, plants are the same. So all of the plants in the environment have fungi, bacteria, archaea, microbes that live in their tissue, on their tissue, in their roots, and they affect plant health. And plant health is important to understand because plants are the root of all ecosystem functioning, and they're also how we eat 3 times a day. So all ecosystem functions in agriculture and the services that we get from ecosystems through food, through fiber, through wood, they're all in turn influenced by the microbes that affect plant health. So they affect plant productivity, pathogen protection, water usage, nutrient uptake, and a whole suite of other things. So that's why I study microbes that live in association with plants. So there are many, many different types of microbes that live inside plants. And I study one group called the Arbuscular Mycorrhizal Fungi, and I will shorten that just to call them AM fungi. And so AM fungi live within plant roots. And you could even say that the very reason we have plants on land is because of these symbiotic fungi. So the very first plants, when they colonized land, hundreds of millions of years ago, plants were all aquatic, and then they colonized land, but they didn't have extensive root systems, so they formed symbioses with these fungi as a way to take nutrients up from a solid environment when they were used to living in a liquid environment. So almost all plants on Earth form symbioses with these fungi. And it's a beneficial association, it's a mutualism. And the fungus takes up nutrients from the soil like nitrogen and phosphorus. And in exchange, the fungus gets plant carbohydrates. So photosynthetically derived carbon that the plant makes, it then gives to the fungus in this sort of exchange. Okay, so this means it's a nutritional symbiosis. And so, like I was just saying, the fungus takes up nutrients from the soil and gives it to the plant, and the plant does photosynthesis and makes carbohydrates and gives it to the fungus. So usually we think of fungi as being decomposers, right? Living on dead things, or maybe being pathogens. Well, these fungi that are incredibly common, exist in all ecosystems, they don't do that at all. So kind of everything you think about how fungi function, it's just not really the way that they commonly function. So these fungi don't form disease, they don't harm the plant, instead they benefit the plant. And they also provide protection against other pathogens, they can provide drought tolerance, and plants can live without these fungi, but it varies, their dependency on the fungal symbiont varies. But the fungus cannot live without the plant. So these mycorrhizal or AM fungi, they have to live in association with plants. And you see them, and they exist everywhere below our feet. So they're in your lawn, they're in forests, they're in parks, they're in urban, in association with urban trees. almost all crop species associate with these fungi, and so they're incredibly important ecologically and also economically. Okay, so even though they're generally beneficial to plants, recent research in the last couple decades has shown that the relationship between these symbiotic fungi, the mycorrhizal fungi, and plants really varies along a continuum from mutualistic to parasitic. So on the left side, you can see that the fungi can really promote plant health in a mutualistic relationship. Sometimes the relationship is commensalistic, meaning they don't harm each other, they don't help each other, but sometimes the fungus can be parasitic on the plant too, in cases where it's not delivering as much benefit as it's getting in return. So sometimes ecologists apply these economic trade models to this association, which is kind of a fun application and a interesting way to understand this natural phenomenon. The relationship between plants and fungi also depends on which fungal partners are present. So in this cartoon, you see two different plant individuals growing, and then they're often found in association with lots of different mycorrhizal fungal species. And so which fungal species are present will really determine how much benefit is conferred to the plant. And we know from a fair amount of research that lots of different factors determine which species you find in a given location or not. And some of those factors can be what we call deterministic or niche-based. So that means like the soil pH or the climate of the region, or even the land history of the region that can affect which fungal species you see in a given location. But increasingly, we're starting to look at stochastic processes, we call them neutral processes, or random processes that affect whether you see a species in a given location or not. And so one of those is dispersal. So dispersal is considered a stochastic process that can drive whether you see a species in a given location or not. Okay, but when we're talking about how microbes disperse, that's a big research gap. So we don't really know how microbes move around the globe very well. And a lot of the field is kind of based on this almost 100-year-old hypothesis now that was proposed by a microbiologist named Bas Becking. And he said, he was Dutch, and he said, everything is everywhere, but the environment selects. In other words, microbial dispersal is unlimited. So microbes can get anywhere and everywhere, and it's really the environment or those niche-based or deterministic factors that predict which microbial species you'll see in a given location. But we've been sort of laboring over this almost 100-year-old hypothesis, and there hasn't been a lot of empirical data to test, well, how limited is microbial dispersal actually? You know, we just haven't been really disproving or generating data in support of this hypothesis in an empirical fashion. Okay, so then bring it into my work and the microbes that I study, these beneficial symbiotic fungi. How did they disperse? And one thing that's also a little uncharacteristic about these symbiotic fungi is they form no mushrooms. So usually we think, okay, a fungus is a mushroom, but the mushroom is really just the fruiting body or the reproductive structure of the fungus. And most of the body of the fungus, I don't know if you can see my cursor, is in these hyphal networks. So these really kind of branched filamentous networks of the fungus, that's the main body of the fungus. And then every once in a while, it'll put a mushroom or a mushroom-like structure above ground to reproduce. But a lot of fungi are cryptic and they don't form these obvious above ground structures. And that's the case of these AM fungi or mycorrhizal fungi. They exist entirely below ground. So you would think that their dispersal is limited, but we're not quite sure. So I've done some work here in Chicago that has indicated that these fungi are able to disperse despite not having above ground structures. We have some preliminary data, well, it was preliminary at the time. We have some data that shows that these fungi can actually disperse into the most surprising environments. So this is my student, Ashlyn Royce. She's an environmental science undergraduate student. And we did a study to sample these symbiotic below ground fungi in green roofs. And we were interested in understanding from an applied perspective whether these fungi are helping green roof plants survive these kind of harsh urban environments. So green roofs, they don't get extra water, they're nutrient stressed, they fluctuate between periods of extreme drought and extreme flood. And so we thought, well, these fungi are so beneficial, maybe they can help green roof plants and promote some of the functions that green roofs provide in cities. So green roofs are put on rooftops to absorb excess stormwater flooding. They insulate buildings to provide less energy usage. They can also serve as urban biodiversity patches for birds or pollinators. And so we thought, well, what's the mycorrhizal link with these green roofs? So we went out and collected a bunch of data, and then we used a statistical modeling technique called structural equation modeling, which allows you to simultaneously test multiple direct and indirect hypotheses simultaneously. And so we built this a conceptual model of sort of the factors in our system and who affects what. And so we thought, okay, well, plant cover will likely affect the amount of fungal propagules in the soil because these are symbiotic fungi, right? They have to have plants. So the more plants you have, likely the more fungi you have. Maybe the plant diversity or the number of different types of plants will affect fungal propagules. Soil nutrients might, roof age might, and then we kind of plugged all of our data into this model, and then we got estimates of the direct and, or I should say the relative contributions of these factors. So there's just a lot of numbers here going on. And let me just call your attention to a few. And the first really interesting pattern that we saw was that when we were looking at factors that have just direct effects on fungal abundance in the soil, really the strongest direct effect was of roof age. So if you look at just the numbers going into fungi here, so we've got 0.5, 0.2, negative 0.19, negative 0.15, and then 0.29. So of all the factors that we measured, the strongest direct effect on fungal abundance came from roof age, which is not what we would have predicted. So, and this kind of indicated to us that fungi in the soil were passively building up in these green roofs. So they're not they're not installed in green roofs initially. It's just the green roofs start with very kind of sterile soil, but they're kind of building up over time over the course of a decade. And, you know, how are they getting there? You know, how are these fungi getting onto, you know, the 50th floor of a skyscraper in the city? And I'm just going to fast forward a little bit. And this kind of highlighted how there are a lot of gaps in our knowledge of how these fungi disperse. So they don't form mushrooms, so how do they get out of the soil, or how do they get out of the rhizosphere? Do all species disperse in similar manners? Do they vary with respect to survival when they're traveling? And these gaps are important not just from an ecological perspective. We want to understand how microbes, how biogeographic patterns in microbes vary, but these are also fungi that farmers and conservation biologists actively manage. So because these fungi are so beneficial, or have been kind of touted as being mostly beneficial, there is, you know, there's multi-million-dollar industries on creating inoculum, bioinoculant with mycorrhizal fungi in them that you can buy and add to your soil and potentially increase plant growth or plant health, you know, nutritional status of the plant. But why should farmers spend hundreds of thousands of dollars on this product if the fungi are just going to disperse into their field passively, automatically? But there just hasn't been much data on this passive reestablishment of fungi. So that's kind of one of the exciting policy or management implications that I think can come from this work. And a couple of years ago, we wrote a policy piece on what are these knowledge gaps that hinder our ability to manage mycorrhizae, and we created this decision tree. In other words, what do you need to know before you consider adding mycorrhizae to your field if you're a farmer or a land manager? So is this inoculum required? Most times people don't know, and maybe you can infer from other ecological properties, but really one of these big questions is whether passive reestablishment of the fungi will occur. And that's kind of what we're getting at now with this new research direction, and you know, why we even study this. Okay, another big component is linking in traits. And this is kind of a big, a new exciting big data direction that a lot of ecologists are taking now, is how to, so, you know, you can measure species in the environment. We can take an environmental sample, amplify all the DNA, and then look at sort of the genetic diversity or the microbial diversity using environmental DNA techniques. And you can understand your site or your plots or wherever you sampled, but how well are you able to predict what you might see in another location? So the predictability in these systems is really, really difficult. So that's where traits come in. So folks are really starting to build databases with traits, plant traits, root traits, fungal traits, microbial traits, to try to be able to predict the behavior of ecological systems based on traits. So in other words, we see a species behaving one way and it has a certain trait, so then can we use traits to predict how ecological systems will function in a different location. So there are a lot of plant, I mean, there are just a lot of ecological trait open databases out there right now. But none of them address this group of fungi, which, you know, it's one of the most widespread groups of fungi, but there's no trait data on it. And it's partly because we just don't, because they have to grow in association with a plant, they can't be cultured easily. And so then our ability to measure their traits in a lab setting is highly limited. So in my lab, we're trying to figure out how to get around this. And one thing we've been honing in on are the traits of these fungal spores. So the fungi, they exist entirely below ground, but they do form these spores below ground that are just born off the end of a hyphal filament. They'll just form a spore. And different species form spores with different morphological traits. And the traits used to be used to identify the species back in the day before we had molecular genetic techniques. But then they kind of started to fall out of fashion as high throughput sequencing became more popular and cheaper. Fewer people were utilizing spore traits. But now they're kind of having a resurgence, which is exciting because I think this trait revolution is contributing to that a little bit. So what do I mean by their traits? So their spores differ with respect to size. So their volume can vary. Some are very small, some are large, quote unquote large. So large meaning, to put it in perspective, like the head of a pin. That's pretty large for fungi. Some of the spores vary with respect to shape. Some are darker, some are lighter. They vary with respect to surface ornamentation, abundance, wall thickness. So some of them form, if this is the spore, they form a really thick wall. And this is an example of a spore with a really thin wall, but it has these projections. And we don't really know what these traits are for. So their biological function, their evolutionary adaptivity, we don't really understand, but I think that's sort of a new direction folks are going. Okay, so in my lab, we kind of combine a lot of these techniques. So we use macroecological approaches. So we do measurements of fungi in the air and in the soil across large spatial scales. So we started off doing this in Chicago and some in deserts in the Southwest. And now we're in the middle of setting up an experiment where we're measuring fungi in the air across all of the United States, 20 sites across the United States, including Alaska and Hawaii and Puerto Rico. And then we combine what we see in the air with sort of remote sensing data that we don't collect, but it's collected by the National Ecological Observatory Network. They have a ton of open data also. And then we combine that with trait-based ecology. So the fungi that we measure in the air, we assess them through DNA-based techniques and high throughput sequencing. And then we also assess their traits and try to make predictions based on traits. And then we do a lot of synthesis too. So we pull together existing data as much as there is, and then also try to contribute to it and synthesize what we know about the fungi and their behavior, their dispersal ecology based on the larger body of existing published data. Okay, so I just want to share some findings that we've had using these approaches. And the study that we did in Chicago was addressing one main question, which was the fungi that we see in the air, do they have specific traits? And are there temporal patterns that can help us inform how these fungi disperse? So we use these dust collectors, and we have put them on rooftops in Chicago. And the way they work is there's sort of a wind vane on the back, and then air travels into an opening of this chamber, and then the top of the top of the chamber has mesh on it, and so the air flows in and up the top, but then when the air slows in velocity when it's in the chamber, particles fall into a settling basin. And so we had those out for an entire calendar year, so starting in January, and we would keep them out for a month, and then we'd collect the samples after every month, and then we would assess the spores using, count the spores under the microscope, and then we also extracted DNA from samples and used high throughput sequencing to determine which fungal species are present. So we're trying to mash together fungal trait data and then also some bioinformatics data too. Just keep an eye on the time here. Okay, and we found a lot of spores, which was surprising that we found the spores for these fungi that form no mushrooms or above ground structures. We found them in all months, even in the winter, even in the middle of snowstorms. We see them flying into the city. We're not quite sure where they come from, but we have some hypotheses. Thousands of spores, lots of different species types, lots of different morphologies, and lots of different sequence-based species or operational taxonomic units is what we call them, since we can't really call them species. This is what they look like underneath the microscope. And then they had lots of different traits. So this is a heat map of the traits of the fungi that we saw over the course of the entire year. And so we could see that the spores that we found were generally small. So we saw some in the larger range, but most of them were lower than 70 microns in diameter. Most of them were smooth. They didn't have ornamentation. We're not really sure why. And they varied with respect to color. So some of them were highly melanized, dark black spores, and some were very light, almost clear see-through spores. So this helps us try to understand, helps us try to get at, what these traits are for and whether the traits affect their movement in the environment. And then we can compare the traits of the fungi that we collected in our samples to the traits of all of these fungi, all species that exist on Earth. And so the fungi that we see in the air in general are smaller in diameter compared to all of the species in this AM fungal group, which is called the Glomeromycotina. And the interesting part was that the aerial dispersal of these fungi was sort of traditionally thought to be pretty rare, but using traits, we could sort of predict that it's probably not as rare as we originally thought, and that likely a third of all described fungal species are able to disperse in this manner. We also saw a pretty strong temporal pattern in when we saw these fungi in the air. And so, we saw them in all months, but there were, some in the winter months and that dipped down in the spring. And then really the end of summer and fall is when we saw a big spike in when we see these fungi in the air. And then it varies a little bit with respect to species. So this species, Atrapii, peaks in August. This other species, Microcarpum, has a clear peak in October. This species might have a spring peak, and another one just pops out only in July. And, it might have to do with when these fungi sporulate, or maybe when they're able to be released from the air, but hard to tell. And then if you look at just at the fungi as a whole, together as a community and kind of like a multivariate space, then the whole community does shift between seasons also. And we see this not only with the spore trait data, but also with the sequence-based data too. And the cool thing about the sequence-based data is that we can compare our sequences generated to taxa that are in these global databases. And so, you know, I could blast this taxa number 193 and see who else has found 193 and what type of environment they have found it in, which is cool because 193 and then 130 and also taxa number 306 were all fungal species that had been previously found in agricultural soils. So that kind of helped us get at a bit of a mechanism for how these fungi are really getting into the air. And so this is leading us to sort of our favorite working hypothesis that the reason we see this peak in fungal abundance in the fall is because that's a time in this region where there's high levels of agricultural soil disturbance. And that may be a mechanism for getting these fungi out of where they live below ground into the air. And that maybe it's a human-based disturbance that is really contributing to the long distance dispersal of these fungi. Then this hypothesis was further supported when we looked at some remote sensing data. So there's a lot of publicly available remote sensing data on when plants senesce. So in other words, when do annual plants start to finish their life cycle? And in Illinois, it's fairly easy to do that because most of the plants growing in our state are either corn or soy. And so there's satellite data that can help us determine when corn and soy are predominantly starting to end their annual life cycle and what time of season that is. And we were able to see that that really coincided when we saw most of the fungal spores in the air. It's kind of nice that not every state has this data, but they use it primarily to help farmers determine when they should harvest their crops. And so the state agricultural survey has this data, and they'll say, okay, it looks like things are starting to senesce, and so you should plan for your crop harvest at this time of year. And so we were able to get this data specifically for the year when we collected the fungal data. And this is helping us really kind of solidify this model for how these fungi disperse and how it's a three-step process of liberation. And we think that liberation is likely a human-driven process because soil disturbance does happen naturally, but it doesn't really happen to the severity and the degree that it happens. Humans are causing more soil disturbance in an extensive and severe way. And then there's some type of transport process. It could be wind, it could be animals, it could be water. And then the final step is the colonization of these fungal spores in a new environment to sort of complete the dispersal process. So our future work is setting up dust collectors in lots of different sites, 20 different sites using the National Ecological Observatory Network. And I just wanted to mention this, especially for you all, is that NEON, as it's called, is collecting data, ecoclimatic data at the 20 sites where we're deploying dust collectors, but also an additional 60 across the US. And it's really an amazing and unprecedented ecological data phenomenon. It doesn't exist anywhere else in the world. And they're online now. The Neon data portal is up and new data products are coming out all the time. They have lots of tutorials for how to access the data, and they have their own utility packages. And so this is something that I do in my ecological data analysis course that I teach for graduate students, is that we do some units on accessing Neon data and playing with it. And there's just a huge potential for new continent scale examinations of ecosystems, just like ripe for the picking. And it's just a matter of familiarity with the data and generating novel questions. Okay, I think I'm going to stop there. Yeah, I think I'll stop there and maybe take some questions.
Thank you, Bala. I have tons of questions, but I'll let students first ask. Yeah.
You mentioned that that for a time, I guess recently, genetic testing has been the most widely, or has become the dominant method of identifying fungi. And then you said that traits were using the kind of the older fashioned trait based is making a comeback. Sorry if I missed the explanation, but do you see why that would be the case?
Yeah, I think the main reason why scientists are incorporating more traits is so they can kind of understand the why, the mechanisms. So in the DNA-based techniques are really good at, you know, being able to say, okay, well, fungi in this area are different from this area, or fungi over, you know, at this point in time vary from the fungal community at this other point in time, but we can't really understand why. And I think linking it to traits helps us get at those mechanisms a little bit better and increases our ability to predict what we would see in the future.
Can you give a concrete example of that?
Yeah, like with the changing climate, we really want to be able to know how fungi might change in the future, right? Or with the changing climate, plant distributions will shift. So we know that plant distributions and animal distributions shift with the changing climate. So things start to warm, and so organisms move northern or move into higher latitudes. And will their associated microbes be able to do the same thing? And so being able to predict how fungal communities have the potential to change in the future is useful.
I see. So for example, like the thickness of the spores might affect its survivability in different environments.
Exactly. So like the thickness, so how much the spore invests in the protective wall, or how much the fungus invests in the protective wall of the spore, can give us an indication of lots of different things that we don't really know. So is it investing in a thick wall so that the spore survives survives traveling through the digestive tract of an animal? Is it investing in the thickness of a wall so that the spore survives UV radiation while it's moving through the upper atmosphere? I mean, we're just not really sure. So those are some those are some experiments that we're starting in my lab, is exposing spores of different traits to different stressors. and seeing how well they survive.
Cool.
Other questions?
Yeah, I have a quick one.
Sure.
So you said you found patterns in kind of the year to year of the spread of these things, and then you kind of use those for recommendations. Have you guys found any evidence where if you, let's say, recommended in 1 year, let's say September to harvest, and then let's say everyone ignored it and drove that and decided to do it either earlier or later, that kind of would then change the recommendation? Like there's an auto correlation between harvest times in a given year and the effects of people either listening to those or ignoring those recommendations?
So the harvest recommendations are not, they're not between year, they're within year.
But if they 1 year ignored, I mean, because I know they're not between year, but does the following or not following of those impact the recommendation for the next year?
Sure. You know, I don't know a lot about what the ag remote sensors recommend and then whether the farmers follow it or not. I'm not sure if they even, I mean, I'm sure they do because the farmers kind of want that and they invest a lot in it. So I'm not really sure how much there's dissent among the farmer community in not following the recommendations.
And would that impact does that change the soil for the next year? So let's say they did it too early and that disturbed the soil too much because it was not optimally harvested. Would that make the sample probably worse next year as well, or does it kind of reset over the winter?
Well, we don't, I wouldn't expect it to reset over the winter. And I think maybe
Bala, we can't hear you. Maybe something got plugged out or...
how's that?
Yeah.
Okay, sorry.
Yeah.
I think I accidentally hit a button.
Right.
Yeah, I think there was maybe a miscommunication because it wasn't that the harvest activity impacted necessarily what fungi were in the air the following year. So maybe I'm not understanding your question.
No, that I think that was the actual, that answer fit my question perfectly. Thank you.
Oh, okay.
Any other questions? So Bala, I think I would like to understand. I mean, you really explained the the scientific, what I would say is the scientific life cycle of the experiments that you are doing. They are fantastic. So how much data are we talking about in collecting the trait data for a single fungi, and when you are doing high throughput, like matching with the high throughput sequence, what do you mean by match? What does match mean itself? So I want to understand how much data are we talking about over here, and then how challenging is the computational sort of aspect of it, which is the matching, and what times does it take to at least get one match even? Yeah.
I have a feeling that what's challenging computationally for me is probably not very challenging for you.
Right. No, but it's the scientist's perspective, right? And
yeah.
What do they like, given the tools that you have, what is like, in, let's say you've collected samples and you've collected 100 samples. Are you getting that sort of the results for those 100 samples, which is the matching part? like fairly quickly, or there are limitations in existing methods? We don't know. I mean, yeah, I think how much data is there for fungi and what exactly are we matching over here? Those would be interesting questions, at least uh, uh, from a computer scientist perspective. Yeah.
Yeah, there are a lot of limitations.
Mm-hmm.
And so the first limitation, like I said, is building these trait databases. So for these fungi, it's a really actually species poor group. So there are 320 species described across the entire world. Now, we know that those species, or what is a described species, is it doesn't capture all of the species that exist in the world, because there's a large number of undescribed species. And because the fungi cannot be cultured, it's a challenge to try to understand how many fungi do we know about versus how many fungi that are out there. So in global biodiversity estimates, we can't go to every corner of the earth and measure all the species that are there. Even if we could see them, we couldn't do that. We can't do that with insects, we can't do that with ants. And these are microscopic organisms. So that's a challenge. But kind of getting back to this group, so there are 320 species, and then what's called a species certainly has a huge amount of genetic variation within it, and so that's then what we call an operational taxonomic unit. So we're not really able to say what a species is, but we just sort of move on with our lives anyways. But then there's high genetic diversity within species, and then of course within populations and isolates. So then, so when we have a dust sample, and we just extract all of the DNA, and then we amplify the DNA of a specific region. And there is a region of the genome that is conserved across all of these fungi, but also varies enough at the species level. So for most fungi, that specific region is the ITS, or the internal transcriber region, internal transcribed region of the RNA gene. So for all fungi, that's the DNA portion that works. For these fungi, that region doesn't work. And so we have to amplify a different region, which is longer. And so then we have trouble aligning sections. So from the bioinformatics can be tricky, because we'll, amplify in both directions of the DNA, but then we can't always align them. Right. And so alignment is challenging. The other thing that's challenging is degradation of the DNA. And then another thing that's really challenging with these fungi is they're what's called heterokaryotic, which means that they have multiple nuclei within a cell that are not identical. So for us, you look at all of the DNA in all of our cells, the cell of my finger, the cell of my my liver, and the DNA will be the same. But these fungi that we work on, they don't form discrete cells. They're sort of these open tubes, and the nuclei are free flowing within them, and then they kind of mix, and they're not identical. So that creates challenges I have a colleague who I visited a while ago, and she calls it assemblomics, like just the whole science of assembling these genomes once they're being sequenced. So that's a hurdle. With this trait database that we're trying to create, we've basically gone into the text of original species descriptions and pulled out measurements of their traits, their size, their wall, etc.
Right.
And, those are decades old. They can't easily be text mined.
Right.
And so we have just been going in by hand and pulling out the data to create this database, because we can't culture them. If we could just culture them, then we could measure and get a good idea of variation in the traits too. But we're having to rely on old taxonomic descriptions.
Right. Creating the database itself would be challenge, even though the database itself is it might be fairly small, but yeah, that creation is the bigger challenge. I see.
The other challenge is working with this neon data. And so I can collect the fungal data, even the sequence data, we can work through that. But then trying to get a handle on all of the other biotic and abiotic potential predictors in these different sites is a challenge to me. So, neon, they just measure anything and everything. And it's great, but it's also, how to pare it down. And so, in other words, like they have, they've got atmospheric data at every site, and, it's being collected instantaneously all the time.
Right. So it's that, the needle in the haystack problem. They basically have the haystack. You got to find your needle over
That's
there.
right. That's exactly right. And so, it's a matter of, just trying to figure out like how to deal with that amount of data. And so the atmospheric data that they collect is all the time, every second or every 5 seconds.
Right.
But then they also have all the plant data. And so, and they're doing plant community assessments, every 2 weeks. And so
how do you find the actual where there is a pattern? So you have to do a lot of analysis, and I'm guessing that the students are downloading a lot of data, doing a lot of analysis, and throwing away much of the data.
That's right. That is right. Yeah, because there's, then there's that balance between, spatial scale and then temporal scale, and where do you draw the line between how much kind of fine tuning you want.
Right. And I just want to sort of say this for some of the students who have attended my talk in the past. We have spoken about data provenance. This is one of the most important application, what Bala is mentioning over here for provenance, that when you finally see a result that Bala was showing in her presentation in a publication, one may wonder where exactly did this data come from on which she's done analysis. Now, Bala may state that, well, the data is coming from neon, but what part of that data has been used exactly? What specific spatial and temporal section has been used? We got to know that, because otherwise, how do I build trust on what Bala has published? So it's
also with neon in particular, like this neon data has been rolling out now for years,
right?
But there haven't been very many papers that have come out from it. Mm-hmm. I think because of people, probably because people like me are not getting together with people like you, and people like me are just paralyzed by the amount of it.
Right, right, right. No, I know some folks at Neon actually, and because of my geoscience collaboration, and
Oh,
I,
nice.
yeah, I know that they've had issues curation, like they've had these typical issues of maintaining the databases and actually that their curation as well as dissemination of the data is not I wouldn't say is top notch like in some other repositories that we see. So yeah, it's a big problem.
So what do you, so if you could identify a gold standard or a better approach, like what other databases or data products would you say would be
No,
good to check
actually,
out?
I wouldn't know. I mean, the part I was involved was, those guys were looking for better approaches for doing curation. And because our work is in provenance, the idea was that, well, we can maintain the provenance given you have an analysis pipeline, for instance. We can maintain for it. But then, there is still a human element that you have to curate. You have to add tags to it. You have to say that, well, this data was used in this kind of analysis and how exactly did it perform over there. Those are kind of sort of human tags that have been attached.
Yeah.
I wouldn't say that I have sort of a silver bullet for them that this is it, but we can ease the process for them. And because we were part of this discussion, I know that their databases are not that well curated as some other databases that I'm aware of. But it's an interesting area. I mean, definitely, especially like how do you get data from publications as well and be able to make that as, because actually a wealth of information is in publications. So how do you bring that into the database itself is a big problem.
Yeah, I've been, a long time ago, I started involvement in a really large plant symbiotic meta-analysis project. I mean, large for us, large for ecologists. And it was, it was started in 2003 where, people in my field were not even yet widely using R, and it was really challenging to be able to collect data. We had 15 people in our group that were all pulling data out of publications for this meta-analysis, and we wanted to be able to collate them simultaneously. So, there was no simultaneous data collection feature. So we had to make our, there's no Google Sheets. So we had to make our own database and website, and we had data entry forms before there was any sort of easily accessible option. And so we started doing that, and it was good. It was very useful. I mean, everything we did is sort of obsolete now.
Right.
But we learned a lot about, kind of to your point, we learned a lot about the pitfalls of pulling data out of existing publications, and how it's incredibly useful, but really difficult too. And I have a student now who's doing a meta-analysis on food nutrition and how fungi affect the nutritional status of foods, and she's just having a lot of trouble because of data reporting and how there's just no standard for that.
Right. Yeah, no, I can sort of point you to some new sort of research that has opened up in these areas, just because I had been keeping track of how exactly this problem of, improving the productivity of scientists. I've been sort of genuinely been interested in improving productivity for scientists. And they have genuine database problems and small data as well as big data problems. And so yeah, I think offline send you a few pointers for that.
Yeah, that'd be great.
Yeah, especially for publications, how you can pull data from publications, which can build the database for you. Yeah. Okay.
I have one more question actually about the data that kind of spawned from that if we have time.
Sure.
So since you're combining a bunch of data sources that obviously probably have different sets of features and sample sizes and such, how often has that kind of been a major obstacle where you feel like there's a type of analysis that would be most applicable to what you're trying to gain insights on, but you're kind of also not able to use that specific type because either the sample size is too small or half of the data is missing a specific column that with your domain knowledge is necessary? It's just how does overall that affect your model selection process or even your analysis type?
I guess it's so common, I just don't even think about it. I guess it does, meaning incongruities between data and missing components are like, that's the standard. And so I don't think I've ever worked in a situation where the data are complete or there's like excellent congruity. So I guess it doesn't get me down too much. We just keep plugging along.
Just kind of the inherent nature of the domain is the data being a little bit tougher to wrangle.
Yeah, I think so. In this trait database, where we're collating spore data for all of the species in this fungal group, one of the issues is that, like I was kind of saying in my talk, so we're focusing on spore data. We have data for trait data for all of the spore-related trait data for all species, which is very exciting. But like I said in my talk, the spore is just like the ephemeral structure, and the main body and function of the fungus are on those hyphal filaments. So that's a problem, right? So we're focusing our analysis and our understanding on where we have data, but it's really not the most biologically relevant portion of the organism. And so we're trying to get at some data that's more relevant to like the actual body of the fungus, but it's limited. And so it's hard to focus a lot of time on and effort on this component of the data that we know will be limited and incomplete, but it's also really important because it reflects it's the real thing that reflects the organism.
I imagine that probably combined with that you said it's hard to replicate these in a lab makes it twice as difficult.
Yeah, that's right. But I think that's a part where the data, we're combining the data knowledge with the biological knowledge is really important.
Thank you.
Quick question about so why is it just fungi in general can't be cultured, or is there some specific property of these ones that?
It's a specific property of these ones. So most fungi, you can culture them. You could take a little petri dish and you put like liquid agar food in the petri dish. And usually we use something called a potato agar, it's kind of like a liquid potato. It's liquid form of carbohydrates. And you put fungi on the plate and they'll grow and they're really happy to. But because these symbiotic fungi have to grow with a plant, they have to, you can't put them on an agar plate and have them grow. They won't do anything. And so we don't really know why. There was kind of an exciting study that came out last year that said that in addition to carbohydrates from plants, the fungi are getting fatty acids from plants. And so then people started making fatty acid cultures, and it sort of started working. And so we're not quite sure why they can't be cultured, but they can't be cultured. You can culture them with living plants. So you can grow a pot in sterile soil with a plant and add some of these mycorrhizal fungi to the living plant, but they're they have a really low success rate, and it's a big hassle. But there are these culture collections around the world where they culture them on living plants, and we work with those.